In this video, I'm joined by Frank David, the founder of Pharmagellan, and we walk through a case study of how to analyze a clinical trial using the framework that Frank developed in his consulting work and he outlined in his recent book here on how to analyze clinical trials. We did a previous video where he walked through his framework, and this one we applied the framework to a real-life example of what to look for when analyzing a clinical study. So Frank knows a lot about this topic. He, in addition to running Pharmagellan, which is a consulting firm that helps biopharma companies realize the value from their pipeline, was previously the co-head of the transactional consulting group at Leerink Partners and was a director of strategy in AstraZeneca's oncology innovative medicines unit. So hopefully you enjoyed this video, and let me know if there's any questions in the comments. Yep. All right. So you want to jump into the case example now?
Right. So I chose a case which comes out of some client work that I did over the last couple of years. So we're going to stay in the acute ischemic stroke area. So just as a brief clinical background, these are patients who are where basically there's been some sort of clot in the cerebral vasculature, and then you have some localized lack of blood flow. So the current standard of care, depending on the anatomy and how long it's been since the patient had their stroke, the clinical findings, et cetera, the possible options, largely speaking, are either anticoagulation, like what we talked about a couple of slides ago, or if you're in one of the large arteries, you can do mechanical thrombectomy. So you can basically reach in there and just try to grab the clot and pull it out. Again, it's somewhat trite to say unmet need, that there's an area with high unmet needs, but let's suffice to say there's an area with high unmet needs. Those two therapies work okay. They work very well for patients who have a witnessed stroke and get to a stroke center very, very quickly and have favorable anatomy and other favorable factors. And as you change any of those parameters, the situation gets a lot worse. One of the holy grails in this area for a long time has been what the field calls neuroprotection, which is this idea that if you imagine cutting off the blood flow to an area of the brain, you have some areas that at any given time that might be irreversibly dead. They were already pretty far away from getting blood flow, and now they've just died off. Then you have some areas that are alive, and then you have this middle region, which you might call the at-risk region, right? They're suffering some ischemic damage, but it's maybe not irreversible yet. And really what you want to do is prevent that from being reversible. Maybe buy yourself some time before you can resolve the ischemia through anticoagulation, for example, and or combat some of the signaling pathways that have already been initiated in those neurons as a sort of an injury response. But this area has been a mess. Nobody really understands the biology of neuroprotection. I mean, this is not news to anyone who's thought about neurology, probably. I don't know that it's any worse than our knowledge of other neuro areas, but let's just suffice to say the neurology is pretty bad. There are a bunch of rodent models. Rodents have very different neuroanatomy and very different neuronal biology. And more importantly, there have been a lot of drugs go through that have looked beautiful in these rodent models, but then have fallen over in the clinic. And there are also no validated surrogate clinical endpoints. So really you end up, it's really hard, if not impossible, to design a phase 2 study that you feel really good about. You can design a phase 2 study, people do, but there are some real problems with surrogacy. So this particular asset, this is this drug called Neuroanatide, also called NA1. The company that owns it is called NoNo, which is a terrible name, but they're a Canadian private biotech. This is their only asset. It's a peptide inhibitor of a scaffolding protein. So essentially, it's supposed to inhibit sort of a neurotoxicity loop that gets set up in response to injury in the neuron. So basically, it's trying to kind of take that to break the self-amplification of the neurotoxicity that eventually will lead to cell death. So the preclinical data are pretty interesting, and I showed it on the bottom. We don't need to go through each of these. Suffice it to say that I mentioned before that the rodent models in stroke stink. There's been increasing use of non-human primate models, which have more similar neuroanatomy to humans. What's also really interesting using the non-human primate models is obviously you can look at endpoints which are actually maybe not exactly how you would assess a stroke patient in the clinic, but awfully close. So actually this non-human primate stroke score, which is the y-axis here, is made up of a bunch of different tasks that are, again, not dissimilar from a neuro exam you would do on a patient in the ICU. And looked pretty good. Again, one could always ask for better, but these, what I can tell you is that when we diligence this with people in the field, there was actually a pretty fair amount of enthusiasm among stroke experts for these types of data. They've disclosed no phase 1 data, and there were no phase 2 data shown either. So, obviously from a regulatory point of view, there must have been some phase 1 studies, but as you probably know, phase 1 studies don't have to be registered in clinicaltrials.gov. So we have no insight into those. And there's a private company, so we have even less insight than we would have normally. And they opted not to do any sort of phase 2 study with their surrogate endpoints. So on the next slide, so we're going to rewind the clock. Oh, sorry, I should just I'll tell you a little bit more about NoNo, which is relevant, again, to sort of overall discussions of valuation and risk. The CEO is a first-time CEO, came straight from academia, actually was the discoverer of Neuroanatide. Overall, I would say from an external perspective, again, I did not do any confidential diligence. This was all non-confidential, just looking at public materials. So I have no insider knowledge here. But from an outside perspective, the team and the board are pretty light in terms of experience and expertise compared to what you would see at the typical Boston or San Francisco area, well-funded biotech by top-tier VCs. So let's go to the next slide. So here's their study. So we're going to rewind the clock to 2016, 2017, and imagine we're looking at this when the study was announced. So they have over 1,100 patients, placebo-controlled 2-arm RCT. That's good. Their primary endpoint is pretty much the standard endpoint, which is the percent of patients with what's called a modified Rankin score between 0 and 2 at 90 days. So the modified Rankin score goes from 0 to 6. 6 is dead. And essentially, this is kind of fits well with sort of what's viewed as clinically relevant and also is a regulatorily relevant endpoint. They didn't disclose their sample size, but if you do a back of the envelope using some of the methods that I alluded to before, it's powered to essentially detect about an 8% improvement if you assume that the placebo arm is going to have about 50% of patients ending up at MRS 0 to 2. And that 50% number, the reason I put that in there is that's kind of what the literature tells you. If you go through the other studies that have been done in this area. Their eligibility criteria, they're looking at patients who are relatively near in, from their stroke. So what the people talk about the last seen well time. So these are people who might have had what's called a wake-up stroke, where they woke up in the morning, they'd gone to bed at 10 p.m. and then at 7 a.m. the next morning, their partner realizes that they've had a stroke, and then they call EMS. So we don't know exactly when the stroke happened, but we know that they were last seen well within 12 hours. Confirmed ischemic stroke on imaging. So the main differential here is with a hemorrhagic stroke. So pretty much every patient gets imaged when they walk in the door, because the management of hemorrhagic stroke is very different. And also, these were all patients who are eligible for mechanical thrombectomy. So as I said before, this is a subset of patients who have these large vessel clots that you're basically going to treat them in conjunction. You're going to treat them pharmacologically in conjunction with a device-based intervention. That's important for two reasons, not to get too far off topic, but from a valuation point of view, that would probably be the label, and that would be the population that you would get approved for in this kind of situation. And that's a relatively small percentage of strokes. Many strokes are in small vessels, distal to the large ones that would be accessible by mechanical thrombectomy. So you're actually going for a relatively narrow population here. And yet you're also creating a somewhat more homogeneous population, because those patients are probably more similar to each other than they would be if you were to expand it.
So that would, having the population restricted to that, patients who are eligible for MT, would potentially make you more confident that the the drug would work, do you think, based on the...
Well, certainly compared to not doing that.
Mm,
That's absolutely right. I mean, if you release either that constraint or the time constraint in general, there's both less chance. What do you worry about? You worry about irreversibility, right? So you worry what percentage of the patients could you ever get to an MRS score of 0 to 2, right? So if you have a bunch of little clots and you can't give mechanical thrombectomy, right, and they're all distal, or if you made it last seen well within 24 hours, those are things where you've basically, you don't know where your, what of the 50% of patients who with current standard of care don't get to MRS 0 to 2, you don't know what percentage of that. You suspect that even fewer of them would be salvageable, would be rescuable in those situations.
mm-hmm.
And it turns out, again, that this is, I would hope it would go without saying, but one of the first things that one really has to do in a situation like this is just really dig into all of the precedent studies that have been done in this clinical area. So what one would find is that there's actually a large number of trials with pretty similar eligibility criteria, and this is viewed as kind of the logical first port of call for a new treatment in acute ischemic stroke. Most of the trials in which they don't have these criteria are of things that have already succeeded in another context, and they're really just trying to expand the utility. So this is kind of the field standard for how to approach this. Certainly, though, I would say if I saw a new molecular entity that was being tested in stroke where the first trials did not have these types of eligibility criteria, I think I'd be pretty concerned.
Mm-hmm. Yeah, it just a lot. You have to have a pretty impressive drug if you're going to go after patients who are that at risk for having
and you have to and you have to just be taking on just a huge amount of additional risk, right? So that's the kind of situation where I might not feel like the Neuroanatide phase 3 has an overwhelmingly great POS, but it's a lot better than it would be if the same study was out to 24 hours and all comers, right? So I think that's really the point that I would make there.
Yep, makes sense.
Good. So again, it's worth just sort of imagining. One of the things that I always, that's a little bit of a pet peeve of mine, is the Monday morning quarterbacking in biotech. So, it's worth going back and putting yourself really at that point before the phase 3 and asking where the main risks are. We don't have to go through all of these, but I think that you could, as the discussion hopefully has shown, you can go through each of these and really start to tick off where are you more confident and where do you feel like there's more risk. I would say in this case, the trial design risk is probably the least problematic, but there are plenty of other, and the data-driven risk is kind of middle, maybe slightly better than usual, given the fact that, if you believe that these, that the non-human primate data are better than the average thing. And then the other areas are challenges, certainly. I'd be looking at this specifically a little bit concerned about, is this worth less in NoNo's hands than it would be in Biogen's hands, for example, is a question worth asking oneself. And then, of course, the inherent scientific risk is huge in this area.
Right. And then with this example overall, was the goal of this evaluation to assess a potential investment into the company, or was there some other goal?
That's a great question. So actually, in this case, this was a company that was developing a strategy for its own stroke program. So we were actually just using this as sort of a competitor benchmarking. What can we learn from them, basically?
Okay.
So it was actually a totally different, the client context was very different in this case.
Okay. Yeah, I was going to ask what the the deliverable or the conclusion is that you have with this information. So
I think that again, let's go through and we'll come back to that because they need some data, right? And we can talk a little bit about that later. So anyway, this trial's done and the results were published in Lancet in 2020. So the top line, it failed on its on the intention to treat population on its primary endpoint. And you see the stats there. But then they did an unplanned post-hoc analysis and they stratified by who got anticoagulation and who didn't. And they found that actually the drug did seem to work in the patients who did not get anticoagulation beforehand. And they went on and they further found that there appeared to be some sort of weird pharmacologic interaction between alteplase, which is the anticoagulant, and Neuroanatide. So essentially, alteplase was inactivating their drug. So they had a mix of patients within their study. It was about 50-50 of patients who had been anticoagulated and those who hadn't. I should have said at the outset that the decision of who gets anticoagulated and who doesn't is based on myriad factors. Some of it has to do with the imaging, some of it has to do with medical comorbidities, et cetera. So, and their study allowed both groups. It allowed people to come in regardless of whether they had been anticoagulated or not. So this was totally unexpected. And then on the good news, the good news was that there was no significant safety signal. I would just point out here, as long as we're talking about evaluating clinical data, obviously this is a post hoc analysis. So, it has to be taken with a grain of salt sort of inherently because, you just worry about multiplicity of testing. That said, it's not like they tested 1 million randomly chosen biomarkers or patient selection criteria and then just happened to find one that worked, right? So in the realm of multiple hypothesis testing, this is probably on the less concerning end. I would also say that the statistical significance here is not that great, right? So it just barely crosses the 95% confidence interval just barely stays to the right of 1. So, going back to that question about sort of what do you think, the overall kind of weaker data, data rarely get better from study to study. Usually there's a reversion to the mean. So, that would be something to sort of keep in mind. But certainly some good plausible, they didn't present all the pharmacologic data, but, having heard discussions of this among KOLs, I think people are pretty convinced by that explanation and that story, that there was some sort of direct pharmacology. It wasn't just, it would have been more worrisome if they just analyzed these two populations and couldn't come up with a plausible reason why the drug worked in one of them and not the other. But here, there at least is a plausible story that they can tell from a scientific point of view, from a pharmacologic point of view.
So you sort of, on the good news side of things, this is not one of 1 million post hoc analyses. It's also, you said it's the large subset, so 50% of patients. So it's not like this tiny little subset. And then I guess sort of on the mixed side, it is statistically significant, but really not super robustly. And then on the negative side, obviously it's a post hoc analysis. Significance is not that high. And then also on the positive side, they have a potential explanation for why you're seeing this effect. So Absolutely. On balance, it's a post hoc analysis. It's not the worst one you've ever seen, but is it convincing enough to run a phase 3 in patients?
That's exactly right. I mean, I would say much better than we got positive results in left-handed redheads, right? So in the whole, again, it's good to set your bar, right? I mean, and you'll see things like that, right? You'll see crazy stuff where, there's all sorts of shenanigans being pulled in terms of phishing, p-hacking for significance by looking at different subsets. And this is sort of the obvious first port of call. In fact, you probably would have had to do that analysis, look at the no alteplase versus alteplase, even if you had gotten the positive top line result. Regulators probably would have wanted that, and probably that was part of the plan, that was almost certainly part of the statistical analysis plan at the get-go. Or what should have been. I mean, it's actually sort of surprising that they claim it was an unplanned post hoc analysis, because I would imagine that usually is required. And certainly after this point, the feedback certainly that I've heard from clinical development and regulatory experts is that since these data, pretty much everybody, most regulators now are saying you should build in a plan to analyze the alteplase versus the no alteplase group if you're going to have them both mixed in your population.
Okay, makes sense.
Good. So now we're putting ourselves back to it's 2020 or sort of early 2021. So now they've decided to run a trial called Escape Next, which is essentially the same as their original trial in terms of size, except now they're just restricting it to non-anticoagulated patients. So now this is a really interesting situation because now, from a Bayesian point of view, how do you consider the risk of this trial, right? So the inherent scientific risk is still reasonably high. The data-driven risk, I would argue, is probably a lot lower because, there's there's something there, and those data are not terrible from the original study, and there's a good reason to think that the hypothesis is correct. The sponsor-dependent risk, you could argue, well, these guys did manage to pull off an 1,100 patient phase 3 trial in stroke, so they're not complete. They're not complete nincompoops. They clearly can do something. So you might actually believe that the sponsor-dependent risk is much less. This is one of those cases because it's private. We don't have good insights into the financials and other types of things. So, there may be some risk there, but it's probably a little bit better than it was before. And again, trial design risk is probably at, even a little bit lower, or certainly no worse than it was for the first study. Interestingly, they also have another study ongoing, which they call Escape Frontier, which involves administering Neuroanatide in the ambulance. Like basically you find the patient, you haven't even imaged to find out whether it's an ischemic or a hemorrhagic stroke, and you give Neuroanatide to those patients. So it might be worth thinking also about sort of how risk tracks across different studies within, different related studies, right? So these are studies with the exact same it's the exact same drug. But this situation also sometimes comes up in platform types of companies where you might have the same underlying technology or the same target being addressed through two different means. And so as you get data from one arm, now you're sort of looking at what the risk is in the other trial, and maybe there's some feedback or sort of feed forward to how you evaluate the POS of the other trial. So again, that would be something interesting to think about as well. And then I think I have one more slide that's a little bit broader. Just, kind of beyond, maybe not somewhat unrelated, but also beyond trial POS is the overall DCF kind of valuation questions, right? So, I think I'd be asking some questions, compared to the original trial, the one that's only going to recruit the no alteplase patients. How's the cost and especially the time going to be different for recruiting that trial? Obviously, you've cut off half of your population. The regulatory implications, you'd imagine that it would be perfectly feasible to get a label for only patients who have not gotten alteplase. But again, you just want to make sure that's really feasible. Pricing and market access is something which we haven't really talked about, but certainly, be thinking about, this is an add-on to branded therapies. And so how big does the effect size need to be in countries like Europe that do real health technology assessments? How big does the effect size need to be as an add-on to mechanical thrombectomy, for example, in this situation to be feasible? Is it better for them from a pricing and market access point of view that they're not adding on to alteplase because now that cost has been stripped out? The market size we talked about a little bit. The other thing I would say about the market is that I didn't mention here is just understanding really what's truly addressable from a clinical point of view. To what extent are you asking, how hard or easy is it going to be to incorporate this into clinical care in a situation where you have to make a decision about alteplase before deciding to give this drug? That would be something that would be worth talking to physicians about. And then finally, there's a commercial capabilities question in terms of, are these guys going to be able to commercialize this themselves, or are they going to have to partner? And if they do try to commercialize themselves, can they really do it at the sort of speed and intensity that you might build into a model if it were owned by Biogen or Pfizer or something like that? The second question, we didn't really, again, beyond our scope here, and honestly, in the client work that I did here, we didn't really talk about it, but came up sort of tangentially. This Escape Frontier study, the extent to which that's actually, is that meaningful at all? Like, do you have to build that? Is that built into your valuation? Is that pure upside? What kind of probability is that? Just a high risk flyer? Are they basically just buying lottery tickets by running that study? Turns out that study's been running for a while, and it's not clear how well it's going in terms of recruiting. And then finally, if this were a public firm, getting back to the baked in thing that we talked about near the top of the session, to what extent, given that if you believe that Escape Next is pretty likely to succeed because the prior data were positive, then you'd have to ask yourself, has the market basically already decided that it's going to succeed? And then I think the related question is whether there's a plausible pharma acquisition target, and if so, at what point? It's in one thing I would say that's interesting in that regard is if you look at different points at which you might be interested in doing an acquisition, you could make an argument for acquiring them now, that basically you understand the risk of the phase 3 and we're going to bake that into our valuation. We're just going to try to buy it. We're going to shoulder that risk. You could make an argument for waiting until those data come through. You could make an argument for waiting until regulatory approval. You could make an argument for waiting until commercial success has been proven. I mean, there's all sorts of different points here at which it could be you could build a plausible case for an acquisition, and the value certainly changes at each of those points. So I think if you're thinking about it from a business development point of view, certainly, and first, if they were a public firm and you were and you were thinking that you were investing in this because you thought they were going to be acquired, it would be interesting to sort of work through what the acquisition case would be at each of those stages.
And then, so from a sort of bringing it back to the question of what's the probability of success and what do we do with this information? I think your point about if this is a public firm, then this is, if it's a big phase 3 like this, it's probably a lead asset. Maybe it's not the only asset, but this is probably going to be something that comprises a large bulk of the public valuation. And then with that information, you can kind of assess based on what you know about the market, what the probability of success might be. And there's questions about how this is going to fit into standard of care and things like that. But you would essentially be given odds of success by the market. And then you can take a look and say, is this really optimistic? Is this really pessimistic? How much do I agree or disagree with this? And then if you disagree a lot, if the market's pricing at 90% and you think it's 50%, right, then you wouldn't invest. But if it's like a 90% probability and you think maybe 80%, it's a little bit less clear that you have a strong signal. So how do you think about?
Well, I mean, obviously the other piece on the valuation side is what you think the size of the prize is at the end, right? Mm-hmm. So you can maybe accept something that has maybe a lower probability of success if you feel like there might be room for upside, if you feel like you've been relatively conservative on your commercial assumptions. So therefore, okay, like I'm coming in a little light on my valuation. But on the other hand, I've also been very bearish on the commercial side. So there's a lot of ways to win here, right? Like my view, this could end up being a good investment, even if it's a little bit riskier, if it ends up being more, the commercial market even ends up being better than what I thought. I think, that's a situation in which what commonly happens is that people are bullish on both of those. Right. So they've baked in a very aggressive set of market assumptions, and also a very aggressive assumption on probability of success. And I think that's where you want to be a little bit more careful, because now you've given yourself very little. The justification is just a lot harder to make. Like so many things you've added to this list of things that have to go right for this to make sense as an investment.
In this project you did you like do any quantification or bounding of the probability of success for this one, or was more of kind of an instructive case for if they're going to design their study, what lessons can they learn here?
Yeah, great question. We did not actually try to put some numbers around the POS. It was really and I'll honestly say that we all do this, right? We all try to put these probability of success numbers on these trials. But at the end of the day, again, these are judgment calls about how to interpret data and how to describe different forms of risk. So, I think in this particular project, we were really trying to break it down in terms of understanding the clinical development risk of both this study and the study that we were thinking about planning, understanding generally what the regulatory risks are in this area and how big or small do we think they are, and how does that play with the trial design, and then what do we think about the commercialization risks. I think what we ended up concluding was that the big risk for a program like this really was just due to the inherent scientific risk, right? So it was just for their particular for Neuroanatide. For our asset, we had also for my client had some additional risk around just lack of good knowledge about their drug's MOA, which further compounded the inherent scientific risk, and also had somewhat less robust preclinical data, although did have some that was kind of in the same ballpark as Neuroanatide did. And I think, and interestingly, this particular client, again, was somewhat inexperienced in this therapeutic area. So I'd say there were some sponsor-driven risks that the organization needed to kind of wrestle with and sort of see whether they, how good they felt about going into this particular area. And do they think that this was, more or less feasible for them as a relatively inexperienced company? Or, given their lack of experience, is stroke just a very scary area to be in, or can they find ways to mitigate by associating with the right KOLs, getting some additional advisors, and really building some more externally sourced expertise.
Right.
So it was really a little bit of everything, but no, in answer to your question, we didn't get all twisted around the axle around was it a 60% or 70% or 80% probability of success.
Right. And then if you're also, a company thinking about pursuing this indication, another angle you can take is, instead of trying to quantify it, is looking at if there's other stuff in stroke getting funded. And if there are things like this program you mentioned where the sponsor risk has been de-risked to a large extent, you have, about as good of data as you could expect. I mean, obviously, if their prior phase 3 was a success, it'd be a different story, but then, that's, it would be in a different position. But even given that, you don't see a ton of investment in this indication. So if you take away pretty much all of the other risks or minimize them, and you're just left with the scientific risk, then the question is that scientific risk, the intrinsic scientific risk in this case, in this indication, even underwritable? If you're a venture or public investor, and if that's the case, then that's another really important consideration in deciding what programs and indications to go after.
Yeah, I think that's right. And I think, it also gets into trying to put your big company hat on while you're looking at the small company. If Biogen or Pfizer or somebody like that is interested in looking at neurology assets, what else are they looking at, and how does this really stack up in that broader land? How does stroke writ large stack up in that broader landscape? And then within stroke, how does this stack up with the other things that are available in stroke? So often it can be, yes, you want a probability of success number to be able to put into a model. But I think it's equally important to try to develop a sense of the relative POS compared to other things that you're likely to be compared to.
Right. Yeah. And that's, I think, on a practical level, really all you can do. So it's really hard to put a number on these things. But if you compare it to others, then you can get a sense of how you stack up, how others might view the asset, and things like that.
Yeah. That might be a good lead into the last couple minutes here.
So actually, this last topic is going to be the subject of another video, which will come out next week. So if you want to hear more about the topic of where to actually find data to use in your probability of success estimates, tune into the video next week and subscribe to the channel to make sure that you see it. If you have any questions on this video, let me know in the comments, and I'll try to get back to you. And again, the link to Frank's book and his blog are in the description for this video, so you should definitely check those out if you want to learn more about this topic.